The electrophysiological study of cord reflexes is a sophisticated and essential diagnostic and research tool in neurophysiology. This study primarily focuses on understanding the functioning of the spinal cord by evaluating the electrical properties of its neural circuits. It provides valuable insights into the functional status of the spinal cord and peripheral nerves, aiding in the diagnosis and management of various neurological conditions. Cord reflexes are automatic, rapid, and involuntary responses to stimuli that involve the spinal cord without the direct involvement of the brain. These reflexes play a crucial role in protecting the body from harm and maintaining homeostasis. Examples include the stretch reflex, such as the knee-jerk reflex, and the withdrawal reflex. Monosynaptic reflexes involve a single synapse between a sensory neuron and a motor neuron. The classic example is the patellar reflex or knee jerk, which helps in maintaining posture. Polysynaptic reflexes involve one or more interneurons between the sensory and motor neurons. These reflexes are more complex and include withdrawal reflexes, such as the flexor reflex. Receptors are specialized nerve endings that detect specific stimuli, for example muscle spindles detecting stretch. Afferent neurons are sensory neurons that transmit the signal from the receptor to the spinal cord. Interneurons are neurons within the spinal cord that relay signals between afferent and efferent neurons present in polysynaptic reflexes. Efferent neurons are motor neurons that carry the response signal from the spinal cord to the effector muscles or glands. Effectors are muscles or glands that carry out the response. Purpose of electrophysiological studies. The primary purposes of electrophysiological studies of cord reflexes include diagnostic, to diagnose and localize neurological disorders, such as spinal cord injuries, neuropathies, and motor neuron diseases. Research, to understand the fundamental mechanisms of spinal cord function, synaptic transmission, and neuroplasticity. Therapeutic monitoring, to monitor the efficacy of treatments and interventions in neurological conditions. Techniques used in electrophysiological studies. Electromyography or EMG involves two main types, surface EMG and intramuscular EMG. Surface EMG is a non-invasive technique that uses electrodes placed on the skin to measure muscle electrical activity. On the other hand, intramuscular EMG is an invasive technique that uses needle electrodes inserted into the muscle to measure motor unit potentials. Nerve conduction studies, or NCS, measure the speed and strength of electrical signals as they travel through peripheral nerves. This helps in assessing the integrity of the peripheral nervous system. Evoked potentials, or EPs, are another crucial technique. Somatosensory evoked potentials, or SSEPs, measure the electrical activity in the brain and spinal cord in response to sensory stimuli. Motor evoked potentials, or MEPs, measure the electrical activity in muscles following direct stimulation of the motor cortex. Reflex testing includes the H-reflex and F-wave. The H-reflex, analogous to the monosynaptic stretch reflex, is elicited by electrical stimulation of a nerve and is used to assess the integrity of the spinal monosynaptic reflex arc. The F-wave is a late response following motor nerve stimulation, used to evaluate the conduction in proximal segments of peripheral nerves. Procedure. Preparation begins with patient history and a physical examination to determine the specific reflexes and areas to be tested. Skin preparation for surface electrodes or sterilization for needle electrodes is also essential. Electrode placement involves placing surface electrodes over the relevant muscles or nerves. For intramuscular EMG, needle electrodes are inserted into the muscle. Stimulation is done by applying electrical stimuli to peripheral nerves using a stimulator. The intensity, duration, and frequency of the stimuli are carefully controlled. Recording the electrical responses is carried out using EMG or other recording devices. The latency, amplitude, and waveform of the responses are analyzed to assess the functionality of the reflex pathways. Interpretation of results. Normal findings indicate intact neural pathways and normal spinal cord function. Abnormal findings may indicate lesions, demyelination, axonal damage, or other pathologies affecting the spinal cord or peripheral nerves. Increased latency suggests slowed nerve conduction, often seen in demyelinating diseases. Decreased amplitude indicates loss of axons or synaptic dysfunction. Absent reflexes may signify severe nerve damage or spinal cord injury. Clinical applications. Electrophysiological studies are crucial in assessing spinal cord injuries to determine the extent and location of damage. They are also instrumental in diagnosing peripheral neuropathies, such as Guillain-Barré syndrome or diabetic neuropathy. In the evaluation of motor neuron diseases, like amyotrophic lateral sclerosis or ALS, these studies provide valuable insights. Radiculopathies involving nerve root compression or injury can also be diagnosed. Additionally, neuromuscular disorders, such as myasthenia gravis or muscular dystrophies, are assessed using these techniques.